This podcast for the oncologist is brought to you by Sanofi Aventis. Frequent mutation of isocitrate dehydrogenase, IDH1 and IDH2, in cholangiocarcinoma identified through broad-based tumor genotyping. Abstract. Cancers of origin in the gallbladder and bile ducts are rarely curable with current modalities of cancer treatment. Our clinical application of broad-based mutational profiling for patients diagnosed with a gastrointestinal malignancy has led to the novel discovery of mutations in the gene encoding isocitrate dehydrogenase 1, IDH1, in tumors from a subset of patients with cholangiocarcinoma. A total of 287 tumors from gastrointestinal cancer patients, biliary tract, colorectal, gastroesophageal, liver, pancreatic, and small intestine carcinoma were tested during routine clinical evaluation for 130 site-specific mutations within 15 cancer genes. Mutations were identified within a number of genes, including KRAS, 35%, TP53, 22%, PIK3CA, 10%, BRAF, 7%, APC, 6%, NRAS, 3%, AKT1, 1%, CTNNB1, 1%, and PTEN, 1%. Although mutations in the metabolic enzyme IDH1 were rare in the other common gastrointestinal malignancies in this series, 2%, they were found in 3 tumors, 25%, of an initial series of 12 biliary tract carcinomas. To better define IDH1 and IDH2 mutational status, an additional 75 gallbladder and bile duct cancers were examined. Combining these cohorts of biliary cancers, mutations in IDH1 and IDH2 were found only in cholangiocarcinomas of intrahepatic origin, 9 of 40, 23%, and in none of the 22 extrahepatic cholangiocarcinomas and none of the 25 gallbladder carcinomas. In an analysis of frozen tissue specimens, IDH1 mutation was associated with highly elevated tissue levels of the enzymatic product 2-hydroxyglutarate. Thus, IDH1 mutation is a molecular feature of cholangiocarcinomas of intrahepatic origin. These findings defined a specific metabolic abnormality in this largely incurable type of gastrointestinal cancer and present a potentially new target for therapy. The Oncologist, 2012, 17, 72-79. Introduction. High-throughput tumor genotyping is becoming an integral part of the initial clinical management of cancer patients. The ability to direct targeted cancer agents based on tumor-specific molecular abnormalities has had a clear and positive impact on the management of patients with non-small cell lung, breast, colon, skin, and gastrointestinal tumors. Evaluating a broad range of key cancer gene mutations across diverse cancers has the potential for identifying new therapeutically relevant mutations. Whereas genetic signatures in colorectal and pancreatic cancers have been well defined, this approach can expand our knowledge of less studied gastrointestinal malignancies, such as biliary tract carcinomas. Biliary tract carcinomas encompass gallbladder carcinoma and bile duct cancer, cholangiocarcinoma. Nearly 12,000 new cases are identified in the U.S. annually, and only 10% of patients present with early-stage disease that can be cured through surgical resection. Even with combination cytotoxic chemotherapy, the prognosis for patients remains poor, and the median survival duration is approximately 1 year. New and effective molecularly targeted therapies are urgently needed, although known mutations in the adenomatous polyposis coli, APC, BRAF, deleted in pancreatic carcinoma locus 4, DPC4, epidermal growth factor receptor, EGFR, Kirsten-RAS, KRAS, phosphatidylinositol 3 kinase, catalytic alpha, PIK3CA, and P53 tumor suppressor TP53 genes in cholangiocarcinoma and/or gallbladder carcinoma may help prioritize patient assignment to clinical trials of new drugs. The full spectrum of relevant mutations across the biliary tract carcinoma subtypes has not been clearly defined. Somatic mutations in the cytoplasmic and peroxisomal isocitrate dehydrogenase gene, IDH1, and its mitochondrial counterpart, IDH2, have been identified through large genomic screens of human leukemias, glioblastomas, and sarcomas. At the same time, experimental studies have implicated these mutations as driver events in oncogenesis. These mutations cause a single amino acid change at a conserved arginine residue within the isocitrate binding site of IDH1, R132, or IDH2, R172, R140, resulting in decreased enzymatic activity for oxidative decarboxylation of isocitrate to alpha-ketoglutarate. Although the oncogenic mechanism is not certain, these mutations confer a novel enzymatic function leading to the NADPH-dependent reduction of alpha-ketoglutarate to the proposed oncometabolite R-2-hydroxyglutarate. Recurrent IDH1 and IDH2 mutations have been identified in a very limited number of tumor types, including approximately 70% of diffuse or secondary gliomas, including diffuse astrocytomas grade 2, anaplastic astrocytomas grade 3, secondary glioblastomas grade 4, oligodendrogliomas grade 2, anaplastic oligodendrogliomas grade 3, oligoastrocytomas grade 2, and anaplastic oligoastrocytomas grade 3. Approximately 20% of acute myelogenous leukemia (AML) cases, and most recently the majority of central and periosteal cartilaginous tumors. Collectively, these findings define a distinct subset of tumor types for which IDH1 and IDH2 mutations may provide new insights into pathogenesis and therapeutic targeting. This study presents the results of mutational profiling of 287 patients across gastrointestinal cancer, identifying for the first time IDH1 mutations in a significant subset of patients with intrahepatic cholangiocarcinoma. Our findings further highlight the important role of molecular diagnostic testing for discovery of novel mutations of diagnostic and therapeutic relevance. Materials and methods. Patients and samples. Tumors from patients at the Massachusetts General Hospital, MGH Cancer Center, who had been diagnosed with a gastrointestinal malignancy, underwent mutational profiling. Subjects were identified and consented for testing of their tumor samples under a protocol with Partners Healthcare Institutional Review Board approval. The initial screening cohort consisted of 180 cases of colorectal adenocarcinoma, 43 cases of pancreatic adenocarcinoma, 32 cases of gastroesophageal adenocarcinoma, 16 cases of hepatocellular carcinoma, 12 cases of biliary tract carcinoma, and 4 cases of small intestine adenocarcinoma. Mutational profiling of biliary tract carcinomas was further investigated retrospectively using an expanded cohort consisting of pathological specimens from 52 additional cases of cholangiocarcinoma and 23 additional cases of gallbladder carcinoma. Cholangiocarcinoma cases were designated as intrahepatic, parahilar, common bile duct, or distal bile duct based on independent review of the clinical records by 2 clinicians. Frozen cholangiocarcinoma tissue specimens used for analysis of the IDH1 and IDH2 enzymatic product were obtained from the MGH Gastrointestinal Tumor Bank by cross-matching against the list of patients studied in the expanded cohort. Genotyping analysis. Cancer gene mutational analysis was performed in a clinical laboratory improvement amendment certified laboratory using formalin-fixed paraffin-embedded tissue. Nucleic acids were extracted, and common cancer gene mutations were evaluated using a custom-applied Biosystems ABI Carlsbad, California, PRISM snapshot multiplex system as previously reported. The addition of AKT1, PE17K, and IDH1 PR132C equals G equals H equals L equals S mutational assays provided screening for 130 somatic mutations across 15 cancer genes. Online Supplemental Table 1. Briefly, genetic regions of interest were polymerase chain reaction co-amplified. Mutational hotspots were then evaluated through a single base extension step in which pools of allele-specific primers annealed adjacent to the target nucleotide. Extension was performed in the presence of dideoxynucleotide bases, each labeled with a distinct reporter fluorophore to identify the extended base. 96 degrees Celsius for 30 seconds, 25 cycles of 96 degrees Celsius for 10 seconds, 50 degrees Celsius for 5 seconds, and 60 degrees Celsius for 30 seconds. Labeled products were separated using an ABI Prism 3730 DNA analyzer, and the data were interpreted with gene mapper analysis software. Life Technologies, Applied Biosystems, Carlsbad, California. For the IDH1 R132X, IDH2 R140X, and IDH2 R172X mutations, the primers used are listed in Online Supplemental Table 2. 2-hydroxyglutarate metabolite detection. Frozen tumor tissue from 5 patients with cholangiocarcinoma was weighed and homogenized in ice-cold 80% volume-per-volume methanol using a 1:10 weight and volume dilution of 100 mg tissue per 1 milliliter buffer. Coveris Adaptive Focused Acoustics, Coveris Incorporated, Woburn, Massachusetts, was used to generate tissue homogenates that were subsequently centrifuged at 4 degrees Celsius for 15 minutes. Supernatants were diluted 1:1 with 100 microliters deionized water containing internal standard. Samples were then analyzed using liquid chromatography mass spectrometry as previously described. Statistical analysis. Gene mutation frequencies among different cancer types were compared using the Fisher's exact test. P-values less than or equal to 0.05 were considered statistically significant. In the analysis of the prospective cohort, we adjusted p-values for multiple statistical tests to control the false discovery rate at 5%. No such adjustment was performed in the analysis of the validation group. Results. Patient characteristics. Tumors from 287 patients diagnosed across a spectrum of different gastrointestinal malignancies underwent cancer gene mutational profiling during the course of clinical management at our institution. The cohort of tumors included colorectal, pancreatic, gastroesophageal, hepatocellular, biliary tract, and small intestine carcinomas. Following discovery of IDH1 mutations in the initial cohort, an additional 52 cases of cholangiocarcinoma and 23 cases of gallbladder carcinoma were genotyped. The characteristics of the patients with biliary tract carcinoma evaluated in this project are listed in Table 2. Mutational profiling of gastrointestinal cancers. A high-throughput tumor genotyping platform, Snapshot, has been developed at our institution to simultaneously query for 130 site-specific mutations distributed across 15 established cancer genes. The frequency of mutations in profiled cancer genes in the initial screen of 287 gastrointestinal cancers is summarized in Table 3. Across the overall group, the most widely mutated oncogene was KRAS, identified in 35% of all cases. Although KRAS mutations prevailed in pancreatic and colorectal cancers, they were infrequent in gastroesophageal cancers. Mutations in the PIK3CA gene that encodes the P110 KDA catalytic subunit of PI3K were found in 10% of all gastrointestinal cancers, with the highest prevalence in colorectal cancers. Oncogene mutations that were identified at low frequencies across multiple gastrointestinal cancer types included BRAF and NRAS, whereas AKT1 and catenin beta-1 (CTNNB1) mutations were exceedingly rare. The snapshot assay provided coverage of only the most common mutation sites within each of the tumor suppressor genes tested. Within this limited scope of coverage, TP53 was the most prevalently and widely mutated tumor suppressor gene, whereas mutations in APC were specific to colorectal cancers. Finally, oncogenic mutations in the tested mutational hotspot regions of EGFR, FLT3, JAK2, KIT, and NOTCH1 were not observed. Consistent with reports that IDH1 and IDH2 gene mutations are rare in tumor types other than gliomas, AML, and cartilaginous tumors, we identified IDH1 mutations in only approximately 2% of the total gastrointestinal cohort. However, although the relative frequency of IDH1 mutations was extremely low in colorectal cancer, 1%, it was unexpectedly found in 3 of 12 biliary tract carcinomas. Detailed analysis of IDH1 and IDH2 mutations in biliary tract carcinoma. To confirm this observation, a larger cohort of biliary tract carcinomas was assembled. In total, we analyzed tumors from 25 patients with gallbladder carcinoma and 62 tumors from patients with cholangiocarcinoma, including 40 intrahepatic cholangiocarcinoma patient samples and 22 extrahepatic cholangiocarcinoma patient samples, perihilar, common bile duct, or distal bile duct origin. There were no significant differences in the age, gender, baseline cancer antigen 19/9 level, or stage of disease among patients with intrahepatic cholangiocarcinoma, extrahepatic cholangiocarcinoma, and gallbladder carcinoma. Data not shown. A more extensive mutational evaluation was performed on these specimens, including evaluations of mutations in IDH2 at codons R172 and R140, which were not assayed in our original panel. Diverse cancer gene mutations were identified in gallbladder carcinomas and cholangiocarcinomas in the total group of tumor samples, as shown in Figure 1 and online supplemental Table 3. The overall mutation frequency was similar in the two kinds of biliary tract tumors. The presence of PIK3CA mutations in gallbladder carcinomas was statistically significant when compared with the mutational profile of cholangiocarcinomas, p equals 0.022, as previously reported. The presence of IDH1 mutations in biliary tract carcinomas, initially identified within the clinical screening cohort of 12 patients, was subsequently confirmed in the expanded cohort totaling 87 patient samples. Most notably, IDH1 was the most frequently mutated gene in cholangiocarcinomas, 8 of 62 samples, 13%, and mutations occurred exclusively in intrahepatic tumors. No such mutations were identified in 25 specimens of gallbladder carcinoma, although this difference did not reach statistical significance, p equals 0.099. The primary IDH1 mutation in cholangiocarcinomas resulted in the codon change PR132C, n equals 5, followed by PR132L, n equals 2, and PR132G, n equals 1, online supplemental table 3. Additionally, an IDH2 PR172W mutation was found in a single cholangiocarcinoma sample. Interestingly, neither the common glioma mutation PR132H of IDH1 nor the common AML mutation at codon R140 of IDH2 was found in cholangiocarcinomas. IDH1 and IDH2 mutations predominate in intrahepatic cholangiocarcinomas. Cholangiocarcinoma is a heterogeneous disease that is classified by the discrete anatomical section of the bile duct in which the tumor arises. Intrahepatic tumors occur in ducts within the liver, whereas extrahepatic tumors include those that arise in the parahilar ducts, the common bile duct, and the distal bile duct. There was a statistical trend, p equals 0.086, for KRAS mutations predominating in extrahepatic versus intrahepatic cholangiocarcinoma samples. Figure 2 and online supplemental table 4. Most strikingly, mutations in IDH1 and IDH2 combined were both exclusive and predominant molecular features of intrahepatic cholangiocarcinoma samples found in 23%, 9 of 40 of cases, p equals 0.042 for IDH1. The clinico-pathological features of the 9 patients whose tumors harbored mutations in IDH1 or IDH2 were reviewed, online supplemental table 5. Of these, 5 patients were male and 4 were female. 6 patients had stage 4 disease, 2 patients had stage 1 disease, and the disease stage of 1 patient was unknown. None of the patients had any known underlying risk factor associated with mutational status, including parasitic infections, primary sclerosing cholangitis, biliary duct cysts, hepatolithiasis, or thorotrast exposure. In addition, there was no association with inflammatory bowel disease, hepatitis C virus, hepatitis B virus, cirrhosis, or alcohol or tobacco use. Pathological review performed on stained tissue sections revealed that 3 samples were well differentiated and 6 were moderately differentiated. All tumors showed well-formed glands embedded within moderate to abundant stromal tissue. 1 tumor focally showed clear cytoplasm, and all tumors lacked both intra- and extracellular mucin. The non-neoplastic liver showed no evidence of cirrhosis in any case. Therefore, within this small group, no unique histologic or clinical features were associated with cholangiocarcinomas harboring IDH1 or IDH2 mutations. 2-hydroxyglutarate accumulation is associated with IDH1 and IDH2 mutations. A known gain-of-function activity of mutant IDH1 and IDH2 protein is the production and subsequent tissue accumulation of the metabolite 2-hydroxyglutarate. This biological effect of IDH1 as well as IDH2 mutations in cholangiocarcinoma patients was evaluated using frozen tissue obtained through surgical resection for 5 of the 62 cohort samples. In 3 wild-type IDH1 and IDH2 samples, analysis found an average of 3.41 micrograms of 2-hydroxyglutarate per gram of tumor tissue, 1.199 microgram per gram, 2.497 microgram per gram, and 6.534 microgram per gram. In a cholangiocarcinoma sample with an IDH1 PR132C mutation, 2-hydroxyglutarate levels were elevated 248-fold above normal, 845.9 micrograms per gram. High accumulation of 2-hydroxyglutarate, 588.5 micrograms per gram, was similarly found in a cholangiocarcinoma sample harboring the IDH2 PR172W mutation. Therefore, gain-of-function activity of 2-hydroxyglutarate production and accumulation is a likely biological consequence of mutant IDH1 as well as IDH2 in cholangiocarcinoma patients. Discussion. This study provides the results of a broad-based clinical tumor genotyping approach that was applied across patients diagnosed with a gastrointestinal malignancy. While interrogating 130 mutations within 15 cancer driver genes using our snapshot platform, at least a single gene mutation was identified in 178 of 287 tumor samples, 62%. We observed that the prevalence of the tested mutations varied significantly across disease subtypes. A primary advantage of applying broad-spectrum genotyping equally across cancer groups is the potential for identifying therapeutically relevant mutations not previously known in a particular cancer type. In our study, novel IDH1 and IDH2 mutations in intrahepatic cholangiocarcinomas were found at high frequency, 9 of 40 tumors, 23%, but they were not found in extrahepatic cholangiocarcinomas, 0 of 22 tumors. The vast majority of patients with cholangiocarcinoma present with advanced stage disease associated with a poor prognosis. Therefore, these observations provide an important first step in assessing whether IDH1 and IDH2 abnormalities can provide a new therapeutic target that can improve clinical management of these patients. Despite the best current therapy for patients with unresectable or metastatic cholangiocarcinoma, the median survival time remains less than 1 year. Recent Phase 2 studies evaluating targeted agents that inhibit EGFR or mitogen-activated protein kinase, extracellular signal-related kinase, have demonstrated some evidence of anti-tumor activity in unselected biliary tract carcinoma patients. Our data suggest that cholangiocarcinomas are also molecularly diverse according to sites within the biliary tract. Across the spectrum of cancer genes represented on our genotyping platform, IDH1 and IDH2 were the most frequently mutated genes in tumors arising in the biliary tract within the liver. Our study therefore extends the list of tumors in which mutations in IDH1 and IDH2 may be the driving event in tumorigenesis, including AML, central and periosteal cartilaginous tumors, and a subset of gliomas, and this may lead to new targeted therapies. Furthermore, the presence of an IDH1 or IDH2 mutation may assist in distinguishing intrahepatic from extrahepatic cholangiocarcinoma at the molecular level. The mechanistic role of mutant IDH1 and IDH2 in tumorigenesis is unclear, but it has most recently been attributed to the gain-of-function activity of R-2-hydroxyglutarate production through the NADPH-dependent reduction of alpha-ketoglutarate. The excessive accumulation of 2-hydroxyglutarate associated with IDH1 and IDH2 mutations inhibits the binding of alpha-ketoglutarate to a number of dehydrogenases that require this substrate for physiological activity. Prolyl hydroxylases that function to hydroxylate and promote the degradation of the hypoxia-inducible factor, HIF1 alpha subunit of the HIF1 transcription factor, are alpha-ketoglutarate dependent, and IDH1 mutant gliomas have been closely associated with induced HIF1 alpha protein levels and target gene expression. Aberrantly hypermethylated CpG islands in gene promoter regions have also been rightly associated with IDH1 and IDH2 mutations in a subset of AML and glioma cases, leading to transcriptional silencing across many genes and impaired differentiation. These effects presumably occur through inhibition of alpha-ketoglutarate-dependent enzymes that control DNA and histone methylation and may therefore promote cancer through epigenetic changes. Inhibition of IDH1 and IDH2 may become an effective strategy for cancer treatment in patients selected through tumor mutational profiling. Conclusion: this study identified for the first time a high frequency of mutations in the isocitrate dehydrogenase genes IDH1 and IDH2 in cholangiocarcinomas, specifically of intrahepatic origin. In 2 cholangiocarcinoma samples tested, these mutations were associated with accumulation of the novel 2-hydroxyglutarate metabolite. The frequency of IDH1 and IDH2 gene mutations was greater than the combined frequency of activating mutations in AKT1, KRAS, NRAS, and BRAF in this cancer subtype, making it the most prevalent drug-targetable gene alteration in intrahepatic cholangiocarcinoma. Acknowledgements. We thank Vanessa Scialaba and Jay Hahn for their technical assistance. We also thank Mohammed Miri for his assistance in tissue procurement from the MGH tissue repository, which was used in the metabolite analysis. This work was supported by internal MGH Cancer Center funds.